Hello friends, in this video I am going to discuss about structure of DNA. In my previous video, I discussed some structural aspects of nucleotide, which is the monomer of deoxyribonucleic acid. We know that long race for the discovery of DNA structure was come to the end when two scientists, James Watson and Francis Crick, announced that they had solved the puzzle in 1953. Actually, both these scientists used X-ray crystallographic studies data of Rosalind Franklin and Young Wilkins for solving this puzzle. Watson and Crick submit their data in the form of paper to one of the most reputed journal of that time, that is Nature Group, and surprisingly Nature accepts the paper without sending to the reviewer. The editor of journal comment that Watson and Crick paper is self-explanatory and accurate, and no other reviewer is working in this field now, so we are accepting the paper without reviewing. For this discovery, Watson and Crick got Nobel Prize in 1962 in medicine and physiology along with Young Wilkins. Unfortunately, Rosalind Franklin died due to cancer, so she is not the member of Nobel Prize winning team. Now in this lecture I am going to discuss with you different forms of the DNA, their properties, and then structure of DNA with the molecular basis. Now we know that inside the eukaryotic cell, DNA is present in a well-organized compartment called nucleus, while in case of prokaryotes, DNA is directly present in cytoplasm because prokaryotic cells do not have well-organized, well-developed nucleus. In both the cases, there are various forms of DNA which is reported by the researcher like A, B, C, D or Z, out of which three forms are well studied. They are A form, B form and Z form of DNA. First of all, I am going to discuss about different properties of these three different type of DNA. First is A form of DNA. The A form of DNA is right-handed. That means its helix sense is right-handed, while B forms of DNA is also right-handed, and Z form of DNA is left-handed. Diameter of A form of DNA is 26 angstrom, while in case of B form it is 20 angstrom, and the diameter of Z form of DNA is 18 angstrom. The thinnest DNA is Z, while the thickest is A form. Then base pair per turn. In case of A DNA, it is approximately 10.7 base pair in a single turn. In case of B, it's 10.4, while in case of Z, it's 12. Here you can see the number of base pair in a single turn of a DNA. In case of B DNA, it is 10.4 base pair in a single turn. Now, pitch per turn. What is pitch per turn? If I draw imaginary axis from the center of DNA, then distance travel in a single turn is nothing but the pitch per turn. Actually, in case of A DNA, it's 24.6 angstrom, while in case of B DNA, it is 33.2 angstrom. Here you can see that the distance traveled by DNA in a single turn is 33.2 angstrom in B form, while Z form travel 45.6 angstrom distance. Then rise per base pair. It is 2.3 angstrom in A DNA, 3.32 angstrom in B DNA, while 3.8 angstrom in Z DNA. Here you can see the distance between the two base pair is 3.32 angstrom in B form of DNA. How we can calculate this rise per base pair? Just divide pitch per turn by base pair per turn. If I divide 33.2 by 10.4, I am going to get value, this value. If I divide 45.6 angstrom by 12, I am going to get 3.8. And if I divide 24.6 by 10.7, I am going to get value 2.3 angstrom, which is the rise per base pair in A form of DNA. Now we come to the major groove. In A form of DNA, major groove is narrow, while in B it is wide, and in Z it is flat. I am going to discuss about major and minor groove at the time of discussing structure. The minor groove in A form is wide, in B form it is narrow, while in Z form it is also a narrow groove. Now we have to consider one thing, that you have to draw structure of DNA parallel. Parallel strands is able to show the major and minor groove. If student draw the structure of DNA like this, that case, it is not possible to mark major or minor group. But if I draw the structure of DNA by drawing both the strands parallel, in that case, major group is clearly visible, while the minor group is also can be marked. Okay, but both these groups are important in characterizing DNA. Actually, the form of DNA which is present in the cell is dependent upon salt concentration, pH of the solution, as well as sugar occurring conformation of that particular DNA. Most frequently found DNA in the living cell is B form of DNA. So in this lecture, I am going to discuss the structure of B form of DNA. We know that DNA having two strands of nucleotides running anti-parallel to one another. One strand starts with the 5' end and end with the 3', while other strand start with the 3' and end with the 5'. Both the strands have number of different type of nucleotide sequences. For example, if the first strand have adenine, which is complementary to the thymine, and in between both these nucleotides, we are going to get two hydrogen bonds. That means adenine always paired with thymine by two hydrogen bonds, while guanine always pair with cytosine by 3 hydrogen bonds. Here you can see guanine pair with the cytosine, by 3 hydrogen bonds. Both these nucleotides, adenine and guanine, is purine, while cytosine and thymine is pyrimidine. So that total number of purine in a DNA is equal to total number of pyrimidines. Ratio become 1:1 when we compare purine number with the pyrimidines. If you want to understand the DNA structure in more detail, you have to consider the molecular structure of the DNA. If I select this segment of the DNA, which having 3 nucleotides on 5' to 3' end and 3 nucleotides on 3' to 5' end, and if I draw the molecular structure of that particular portion of the DNA, it will look like this. In this structure, you can clearly note the 5' end of the first strand, and this end will be 3'. Here you can note 3' hydroxyl end of the first strand. This is the first strand of the DNA. Now the second strand start with 3' end, 3' OH. Here you can see, while it end with 5' phosphate. Okay, so the first nucleotide, that is adenine, you can see here. It is a purine, so it is a double ring structure attached to the pentose sugar by glycosylic bond. This is glycosylic bond. In my previous video, I discussed in detail about structure of nucleotide. Okay, so this phosphate plus pentose sugar plus adenine is nothing but the single nucleotide. Now this adenine bond for 2 hydrogen bond with the opposite thymine nucleotide. Here you can see thymine, which is attached to the pentose sugar, and in between adenine and thymine 2 hydrogen bonds are present. Now second is guanine. Here you can see guanine, and in front of guanine on the opposite strand there is a cytosine. In between guanine and cytosine you can notice 3 hydrogen bonds. Okay, and last is thymine. In front of thymine there is adenine, and in between these two there are 2 hydrogen bonds. Okay, at the time of study DNA structure, you have to remember that DNA molecule gets stability due to this hydrogen bond, which held 2 strands of the DNA close enough. In DNA, there are 3 important bonds you have to remember. First is glycosylic bond. This bond held nucleotide attached to the pentose sugar. So in between pentose sugar and nucleotide, glycosylic bond is present. In between 2 nucleotides of opposite strand, second bond or hydrogen bonds are present. While 1 nucleotide pentose sugar attached to the other nucleotide by a phosphate, and this bond is called phosphodiester bond. So in the DNA, 3 bonds play important role in stabilizing the structure. First is glycosylic bond, second is hydrogen bond, and third is phosphodiester bond. When you consider the chain of polynucleotide, one end of the chain have 3 phosphate at 5th carbon of the sugar, So it is called as 5' phosphate end. While the lower end of the same chain have 3' OH or third carbon of the sugar attached to the hydroxyl group. So we call as 3' hydroxyl end or 3' OH end. In this way, if you consider the molecular structure of the DNA, you are going to get the correct or clear idea about the different structural aspects of the DNA. So in my previous two videos and in this video I discuss about the discovery of DNA as a genetic material, then structure of nucleotide, and in this video I briefly discuss about the structure of DNA. From my next video I am going to start with the central dogma of molecular biology, in that replication, transcription and translation process. Thank you.